Figure 8 shows a method of producing plants. So this is cloning through cuttings. The cells are taken from the parent plant. Cells are placed on agar growth medium. That's just a nutrient jelly made from seaweed, which cells love. Cells develop into tiny plantlets, and plantlets grow in compost. Some cells in each plantlet develop into root cells. Name the process occurring as these cells develop into root cells. So basically, when we're talking about cells becoming specialized, the process is called, when they have a specific function, like roots have a specific function to absorb water, we call that process differentiation. Describe the advantages of producing plants by the method shown in figure 8. So you can see they're producing many very quickly. It's quite cost effective. You can do it in a small space. So there's a number of advantages. You only have to describe 2. So produce many plants will do, quicker than sexual reproduction, produce clones with desired characteristics, conserve endangered plants. There's lots of sort of reasons to do it. An autoclave is used to prepare the agar growth medium used in step 2. An autoclave is a machine that uses very hot air and high pressure to sterilize or kill bacteria. Explain why the agar growth medium is autoclaved. Well, it's really important that you understand that these plantlets are very sensitive and can easily be destroyed by a fungal infection or bacterial infection. So we need to protect them. So sterilization is basically a way of killing any other bacteria. So sterilizes agar growth medium. That's why we use an autoclave. So there's no contamination. There's no foreign bacteria, fungi that could destroy the plants. Next, give one of the plantlets. If one of the plantlets had different colored leaves, give one reason why this plantlet had different colored leaves. So we're looking at possible reasons. So it could be environmental reasons or genetic reasons. It could be a mutation, or it could be to do with different nutrients. It could be a number of reasons. So choose a suitable environmental or genetic reason, like mutation or a disease. Crop plants provide a source of energy in the form of carbohydrates such as starch and sugars. Describe the test to identify starch. Starch is detected by iodine, which goes from brown to a bluish black. So that's the test described, basically. Add iodine and turns from brown to blue-black in the presence of starch. 2 easy marks. The amount of energy in the sugars extracted from crop plants can be measured using the calorimeter shown in figure 9. So this is basically when we see how much burning food or heating food can change the temperature of water, because changing that chemical energy into heat energy is a way of measuring the initial amount of chemical energy in there, because energy is energy. We're just transferring one form to another. So what we've got here is the food here, which will be heated, and then the water around, which will receive the heat and will heat up as a result, and will measure the temperature of the water. Now, it's really important that we minimize heat loss, so all that heat is transferred to the water. It's almost impossible to do this perfectly, but we can be pretty good at insulating it as much as we can. So have a look at some of the features of a calorimeter. Firstly, we have this stirrer, which will swirl to make sure the heat is evenly distributed around the calorimeter, so the temperature taken at any point would be the same. Secondly, obviously we've got our ignition wires here, which send an electrical charge which will heat or burn the plant sugars. We have a lid and an insulated coat which will keep the heat in. So explain why the calorimeter has a lid, give a reason, so say what's there for and why we need to do that. As an insulator to reduce heat loss, so the result is more accurate. So that's why it's there. It's important that it limits heat loss, but also that gives us a much more accurate reading of the energy transferred. Remember, 2-mark questions require 2 points. Next, state why it is important to stir the water in the calorimeter. Well, I've just talked about this, to evenly distribute heat throughout the water.